late now, so it's late to you can't teach an old dog new tricks. So they say. You can. It's hard, it's hard. Same with humans. Like.
It's actually now that he's getting older.
Sí, ya empieza a hablar. Que pasan al mar. De emisiones que va y el invierno la temperatura baja 6-7 grados.
Welcome everybody to today's seminar. It's our big pleasure to welcome the first PhD candidate speaking in our seminar this season. It's Adrián Monreal and Nerea Montes. And we will go with Adrián first. It will be a 20 minutes talk followed by a 10 minutes discussion, and then the same for Nerea. And before we start, just a friendly reminder to all of us that we mute our phones. And with that, Adrián, the stage is yours. Good luck.
Okay, thank you everyone for coming. My name is Adrián, and I'm going to present my thesis plan, which is about ecological interactions between tadpoles, anuran tadpoles, and aquatic macrophytes. And it's supervised by Iván Gómez-Mestre and Carmen Díaz-Paniagua. Okay, both aquatic tadpoles and both tadpoles and aquatic macrophytes are. I don't know if I can get this. Okay. Both tadpoles and aquatic macrophytes are key components in freshwater systems, and they have important ecological roles. In the case of tadpoles, they are usually primary consumers and also prey for different organisms. They can reach really high densities. And amphibians are the most dangerous vertebrate group in the world, so understanding their role in this ecosystem functioning may us understand how the decrease in their abundances may affect these systems. Okay, so we know that tadpoles can impact nutrient cycling because, for example, they consume leaf litter and they enhance its decomposition. They also They also impact algal communities because they feed on both phytoplankton and periphyton. Periphyton are the algae which grow attached to different surfaces like rocks or plants. They also impact zooplankton because they can feed on zooplankton, but they can also have indirect effects as they may compete with the zooplankton. Regarding the aquatic macrophytes, when they are present, they are the main primary producers. They also can reduce water turbidity and reduce algal abundances because they also compete with phytoplankton and periphyton. And finally, they increase and they create more habitat, more structural complexity to these habitats. Nevertheless, despite being so important ecologically, the interaction between these two groups or these two kind of organisms has received little attention. Some studies have focused on how plants impact on tadpoles. Usually, they influence tadpoles by changing different variables of their habitat, for example, changing the water physicochemistry or the productivity. Tadpoles can be also negatively affected by tannins or different plant substances. And regarding the tadpoles, they can impact the plants, for example, by herbivory. For example, Pelobates cutriped tadpoles decrease the aquatic plant biomass present in temporary ponds. They also have an impact on plant phenology because they reduce plant flowering period. And it seems that there are some species of tadpoles which may facilitate the plant growth. This may be an effect of the fact that as tadpoles can feed on periphyton, they may clean the plant surfaces, facilitating the photosynthesis. Another interaction between tadpoles and aquatic plants, which was described by Rosa Rivas, is that tadpoles can carry viable seeds inside them. She studied the feces of tadpoles from Doñana, and she found that most of tadpoles carry seeds of the most frequent macrophytes of Doñana temporary ponds. So, knowing all of this, for my PhD, we want to address three main objectives. The first one is to evaluate how different species of tadpoles affect the biological communities of temporary ponds, focusing on aquatic plants, but also we will try to test if they have also effects on water physicochemistry or on zooplankton communities. Also, a second objective, we want to address if the tadpoles affect the seed germination and which kind of role can they have in seed dispersion. And finally, we want to explore if this pattern of this phenomenon that was found in Doñana is also found in other areas of the world. Okay, so now I'm going to explain in more detail each of these objectives. For the first one, we wanted to test the effect of different species, but focusing on the whole larval period. So for that, we have conducted a mesocosm experiment using 90-liter mesocosm, 90-liter tanks, in which we added sediment of 2 temporary ponds of Doñana in order to have a representation of the seed bank and zooplankton egg bank of these ponds. We have studied 7 different tadpole species, which are the most frequent tadpole species in Doñana. These species are ecologically very different. For example, they are different in their trophic ecology, or they are very different, for example, in their size. So some tadpoles, like the tadpoles from Pelobates, are really big, and others are smaller, like Pidalea calamita or Discolosus galganoi tadpoles. So we decided to test similar tadpole biomasses. So we have decided to test the effect of similar biomass of tadpoles in plants. So that's why we have added different different number of tadpoles for each tank. So less tadpoles of the bigger species and more tadpoles in the smaller ones. They also differ in their reproductive phenology. That means that they start breeding at different times. They also end the metamorphosis at different times. So comparing between species is quite tricky, And that's why we have decided to have a set of control tanks. A set of control tanks. And what's this? Sorry. How can I remove this? Okay, it's fine. We have decided to have a set of control tanks and treatment tanks, tanks with tadpoles, for each species, as we want to collect the data once the metamorphosis has started, and each species will start the metamorphosis at different times. So having 7 replicates for each treatment, that means that we had to have 98 tanks. And we decided that the metamorphosis had started once we had 1 metamorph in each tank of an species. So then we will collect the data. We took different measurements and different samples. For example, we measured different parameters of the physicochemistry like turbidity, pH, conductivity, and dissolved oxygen. We have also measured the concentration of chlorophyll of the phytoplankton, that is the water column, and the periphyton. We have also taken some samples of zooplankton after filtering 10 liters of water from each tank, and of course the plant biomass. So we did weight the fresh weight of every plant species present in the tanks. We have also collected some sediment for trying to characterize using molecular methods the bacterial community, but we have to think how to do it, and it's not sure that we are going to analyze that too. Okay, so now I'm going to show some preliminary results regarding the physicochemistry and the plant biomass. Regarding the physicochemistry, the tadpoles did not affect conductivity or pH, but some species did affect turbidity and oxygen. For example, in the case of turbidity, most of the species tend to increase it, but only 2 of them had a significant impact. So Pelodites ibericus and Pelobates cultripes, these 2 species, increase the turbidity of the water. And they also decrease the concentration of dissolved oxygen. And another species, Colosus galganoi, had a similar effect. This figure is the effect size because I'm plotting how the tanks with tadpoles differ from the control tanks. So, I mean, it's not like to compare between species, but just within the tanks of a given species. Okay, Enric. Oh no. Why? Oh, I forgot one. Sorry, I've lost a slide. Yeah, I've lost a slide. So the next slide was similar to this, but with the total plant biomass, and we could have seen that if we measure total plant biomass, most of the species didn't have a significant effect, but again, Pelobates cultripes and Pelodites ibericus, they had a negative effect. So that is that they had eaten the vegetation. And in the case of Pelobates, this effect was quite big, so it was like around the -5 because they have eaten all the plants. I mean, when these tadpoles are quite big, were in the tanks, there was no, even a small branch was left. They have eaten everything. And the next thing is that if we focus on how has changed the species composition of the tanks, first of all, we must take into account that the different species have been sampled at different times, and the vegetation and the plant community of the tanks is quite dynamic, so it changes a lot of the time. So that's why these are the control tanks, and you can see that the proportion of the different plant species that we can find in them changes along the time. So for knowing how the tadpoles have affected this species composition, we decided to do a composition analysis. That means that we wanted to know if the composition of the species, the relative weight of each species, was different between the treatment and the control tanks. Instead of comparing each species separately, we just wanted to know if the composition was significantly different. For that, we did a linear model. In order to analyze this data, we needed to have an isometric log ratio to transform this relative data to do the linear model. And what we found is that 5 species significantly affected the plant composition. So we can say that for these 5 species, the proportion of plants were different when tadpoles were present and were not. And the most common pattern was that the calitriche, the proportion of calitriche, is a plant species, tend to be lower when tadpoles were present. And we think that is because calitriche is a very thin and fragile plant, and we think that is quite palatable and easily eaten by the tadpoles. There are also other changes. For example, in the case of Pellites evericus, the proportion of potamogeton and also the overall biomass was higher. So it's like the potamogeton for Pellites potamogeton has been benefited of the presence of this species. Okay, and just as a summary or a conclusion from these still preliminary results, we can say that the effect of the tadpoles on the vegetation is strongly species dependent, and it may be like a very strong effect, like in the case of Pelobates, which eats all the vegetation, or it can be like no clear effect, like Alites, which doesn't change the plant composition, or that this doesn't change the plant biomass. Okay, and now regarding the second objective, where we wanted to know the role of tadpoles in plant germination and dispersal. First of all, we want to describe which plants are being carried by tadpoles in Doñana temporary ponds, and for that, we are planning to sample tadpoles of the 7 main tadpole species that are present in Doñana temporary ponds, collect their feces, and inspect them under a binocular microscope in order to search for the seeds. And if we found any seed, we will do a germination trial to see if these seeds are viable. Once we know which are the species more frequently transported, we want to know if the ingestion by the tadpoles changes its germination. This is important because in temporary systems, in temporary freshwater systems, plant seeds have a dormancy in order to be able to survive to the dry period. So each generation, different seeds can germinate in different years. I mean, one seed that is produced this year may germinate the following year, or maybe 2 years after. So if the tadpoles change the pattern of the germination, they may have an important role in the plant population dynamic. So that's why we are going to compare the germination of control seeds that have not been ingested by tadpoles, and we want to feed some tadpoles with seeds and test also the germination of those seeds. Tadpoles may affect the germination patterns. They may disperse the seeds within a water body, but they will not provide longer dispersion distances. That's why we also want to address if there is a potential role of secondary seed dispersal if those tadpoles are also eaten by a bird. So we plan to feed also captive birds with tadpoles that have been fed with seeds, and again see how is the germination of those seeds. And lastly, the last objective was to see if this pattern found in Doñana is also found in other areas. We will try to identify if we found any seed from other country. We will try to identify them, and we will do it. First, we have contacted several collaborators from different areas. We have told them to send us samples after collecting them following a standardized protocol. So once we receive a sample, we simulate a dry period. Then we fluid the sample, and we do the germination trial, and we wait and check daily if we can see any seed line from those samples. And after 2 months, we process those samples under the binocular microscope in order to see if there is any seed that has not germinated. And to identify this plant taxa, we are planning to do it by extracting the DNA of the plants and using 2 different molecular markers, one from the gene of the rubisco and the PPSA. And they have around 200. I mean, we think that with these 2 markers will be enough to have a broad option to identify the plants. Okay. And for now, we have contacted several collaborators from different places of the world, and we have already received samples from Colombia, Croatia, the US, China, or Romania, and also Colombia. And the samples from Colombia collected by Alexandra Delgadillo were samples from Dendrosophus molitor And we were greatly surprised because, I mean, it was our hypothesis, but we found more than 100 seeds in those samples in 22 samples, indeed, 22 feces of tadpoles. So there were seeds in 86% of those samples, so it's quite a good news. And just as a summary, regarding the first objective, now we are analyzing the data of the experiment. For the second one, we are planning to start sampling the tadpoles this season. And the third objective, we are still contacting more collaborators, and we are processing the first samples. So finally, I must thank, of course, my group for supporting and helping me every day. And also thank you all for your attention, and any suggestion, I will be glad to listen to it and to answer any question too. Thank you so much.
Okay, so any question for Adrián? I'm sure there should be many of them. Yeah, sorry.
Hello, Adrián. I'm very interested in the dispersal part. And do you think the tadpole can transport, except for seeds, they can also transport invertebrate eggs?
Yes, I think so. Indeed, I mean, I think so because many tadpoles are benthic and they feed on the sediment, so they could eat the eggs that are in the sediment. So yeah, okay. I think this is a possibility. As we are planning to inspect the feces, if I see something that could be an egg, I could tell you.
And do the tadpole eat zooplankton in the field?
In the case of Doñana, I'm not sure, but there are other studies for example, from the US that some tadpoles eat the zooplankton, yes. I think probably, for example, pelobates, which is a quite big tadpole and it eats a lot of things, I think they probably eat also zooplankton.
Okay, thank you.
Okay, one technical question. Are you going to identify yourself the seeds coming from feces, from tadpoles, from other parts of the world?
The seed?
The identification of the plants, are you going to do it? Seems quite complex, no? In Colombia, you said that you found lots of different seeds and...
Yes, so the idea is to identify the seedlings by molecular methods, and we have asked the LEM for help to do the extractions, and yes. And once we have the results of the extractions, the idea is to compare with sequences from GenBank, and yeah. I mean, not morphologically, because that would be very, very difficult. Like having literature for all the world. No.
Yeah, but I guess that will imply also having a good reference collection of candidate species. That is going to be a lot of work in addition to catching tadpoles, I guess. No?
Could be, and I also don't know how precise can be the identification because we don't know how much information about species from other places is going to be. Yeah.
Oh, thank you, Adrián. Very interesting project, and too much worry anyway. But yes, I wanted to precise or to clarify some points of the first part of your project. Regarding, you are basing your analysis of the effect of tadpoles based on single species studies.
Yes.
It's true. Yeah. But do you plan any way to analyze the community effect? You know, in the 80s, 70s, 80s, start a lot of studies of ecological communities using tadpoles of different species. There are great tradition of that using mesocosm. But I don't know if you are preparing some design to see whether interactive effects between tadpoles also affect the output on the chemical, etc.
We are not planning to do that because first, Rosa Rivas, which I was thinking her work, she actually did an experiment with the whole community of amphibians from Doñana. These 7 species, some species were not present in her work, but she studied, I think, the community of 5 anuran tadpoles and also 1 newt. And she did that. She studied the effect on zooplankton, on plants, and on physicochemistry. And she had found that, well, she studied the whole community and also most of the anuran species, but without pelobates. And she had found that when pelobates were not present, the plant biomass had increased respect to the controls. That's why I was saying that some species may have a positive effect. So for this work, we wanted to disentangle the specific role of each species. So we wanted to address specifically species by species. It's true that it would be interesting, like, to start to combine interactions between 2 species and 3, but we are not planning to do that. Yeah.
You can introduce many aspects, of course. Not very interesting. Anyway, the question is relating to the part 3. Which is your expectation of the geographical analysis? What I mean is, you expect in the tropical science, you are expecting that tadpoles are more dispersed, the diversity of species. I don't know. Which are your expectations?
It's quite difficult because we are receiving the data of people who wants to help us, so we cannot focus on specific sites. So we don't know how much detail or how different are going to be the different places, also different species. For me, the most interesting thing would be to know more or less the trophic ecology of the tadpoles and try to link that to their ability to disperse seed. For example, if tadpoles that feed on plants, I would expect that they would disperse more seeds because they could eat them from the plant.
Yeah, it's very nice because recently I read a paper published by a Japanese team that there are frogs that breed in bromeliads, in Phytodermata. No bromelia, Phytodermata in Japan or Taiwan, I'm not sure. But these tadpoles don't defecate at all, so they cannot be dispersed anyway. But you know, the diversity of possibilities are enormous.
Yeah, that way it's hard to have like a very specific prediction. Is objective. Okay. Yeah.
Thank you. Thank you very much.
More questions? Yeah.
Thank you for the great talk. I was wondering regarding not the last one, the question before. Like, I understand that you're like analyzing one species at a time because it would be too complicated to create all the possible compositions. But did you think, like, I don't know how many of these species, like, how's the succession of the species that live in the ponds? Because maybe the previous tadpole species has left the pond in a condition which is not the one you have for the control because they've been eating a lot of a type of plant. Do you think that's something that might be happening, like the starting condition of one tadpole species, which is later in the succession? Would it be affected by the previous occupants in your experience observing how they use the resources in that sense?
Yes, I mean, I think that happens for sure. But also another interesting thing of this study system is that it's very variable between years. So depending on when it starts to rain, the ponds may start filling on November, for example, or in March. And depending on the length of the hydro period, also the interaction of the species will vary because in wet years, maybe the early breeders are at the same time in the pond as the late breeders. But when the hydro period is shorter, most of the species are going to be at the same time. So it's not just that a species may be influenced by the effect that a species had before, but that depending on the years, they may be at the same time in the pond or not. So it's I think it's very interesting to study that, but it's also methodologically it's difficult because you have to decide in which specific scenario you want to focus. But I mean, for sure, in the case of the species that change a lot the habitat, that's for example Pelobates, and Pelobates indeed early breeder, for sure it will have an impact of the species who will breed later on.
Okay, cool, thank you.
Okay, thank you for the questions. If you have more questions, I'm sure that Adrián will be around. So thank you, Adrián, for your talk.
Thank you.
And it's Nerea's time. So let's continue with our next speaker, Nerea Montes. She's working in the Nacho Bartomeu's lab, and she's going to present her PhD. So the floor is yours.
Thank you. Thank you very much for the introduction, and thank you all for coming today. I want to present you my thesis project that is titled Plant Pollinator Networks as Dynamic Systems, introducing the concept of open communities rewiring and their implications in a context of climate change. And to start with, first, the main characters, which are plants and pollinators. In Spain, we're lucky to have a great diversity of both groups, but specifically talking about pollinators, we can find not only wild bees, but also syrphids, butterflies, beetles, and flies. So before talking about the plan of the thesis and the chapters that we have thought about, I want to introduce three key concepts that I want to explain in this thesis. There are plant-pollinator interaction networks, open communities, and rewiring. So starting with the first one, plant-pollinator interaction networks, I'm sure that plenty of you are familiar with this type of interactions. These interactions are composed by a set of plant species, a set of pollinator species, and the interactions that they form between them. They're really important for the ecosystem function of pollination. And usually when we read about these communities in the literature, they're considered closed systems. This means that there's no change or variation in the set of species that they are formed. So that's why we want to introduce the concept of open communities. So when we have our study community here, what we are going to study is how the pool of species at the regional level can also feed this site community that we are studying. And lastly, the rewiring. So having our plant-pollinator interaction network example here, rewiring will happen when a factor such as land use change, climate change, or a new species getting into the system changes one of these links, removes it, and then we find a new one. This is what we will understand as rewiring. And lastly, again, another concept that most of us are aware of, climate change. So just to give you a brief example of the Almonte area, the data that we're working with in terms of climate, we have seen an increase in temperature over the past 80 years, and on the lower plot, we have seen a decrease in precipitation in the past 80 years, showing that we are entering a water scarcity period. So let's go back to the title, introducing the concept of open communities, rewiring, and their implications in a context of climate change. And to tackle this title, we planned 4 chapters. The first one that I'm going to explain, it's almost ready to send for revision. And the 3 next ones are still a work in progress. So let's start with the first one. And we want to answer the question, is biodiversity buffering phenological changes of plant and pollinator communities? And to do so, we planned 3 main objectives at 3 different levels, ecological levels. So the first one is focused at the species level. So we know that species change their phenology due to climate change, but usually the studies on pollinators are focused in temperate climates or even alpine climates. So we want to study how the pollinators and plants are changing their phenologies in a Mediterranean climate, how the climate change is affecting over time the phenologies. So we expect that they will advance their phenologies and shorten their phenologies. The second one is the interaction level. So we expect that there's a change at the species level. So how is it going to affect the interactions? Are we going to see a decrease in the overlap, so the days that 2 species that interact coexist, or not? So we expect that if indeed the phenologies advanced and they shorten, we're going to see a decrease in the overlap between those species. And lastly, so we expect there's a change in the phenology of a species. We expect there's going to be a reduction in the total overlap. So can the community buffer these impacts of climate change? If we think about a set of species that we might find in a community, can the variation in phenologies in all this community buffer the possible overlap loss? So completing those gaps that we might find. To do this, we are working with a dataset that Curro Molina has collected over the past 10 years. We're going to use only 8 years of those, between 2015 to 2022, and within 17 sites, with 9 to 12 rounds per year, always sampling under Mediterranean pine forest and shrublands. Also in this study, we're considering just 17 pollinators and 20 plant species because we need a certain abundance of records of each species to calculate accurately the phenology of each species. In terms of data processing, we calculated 3 different data added to this to the raw data we have. So first of all, the climate variables, we calculated 32 climate variables ranging from precipitation, temperature, and soil moisture. And in terms of phenophases, we calculated 5 phenophases, but we're only using 2 in the analysis. That is the start of phenology or emergence, and the length of the phenology, and in this case will be the flying period for pollinators. Additionally, as I spoke about potential overlap, so we calculated potential overlap, and the first one will be potential pairwise interaction overlap. That is the number of days that 2 species that interact overlap their phenologies. And secondly, we calculated potential total overlap per species. There is the total number of days that one species overlap its phenology with all its partners. In terms of analysis, we performed 3 different analyses for the 3 different objectives. The 2 first are at the year level, so we put by site. And the last one, we wanted to consider biodiversity, so I'll explain later, we didn't pull by site. So the first one, phenophases shifts due to climate change. So first we study how phenophases are changing in over time, so if there's a trend of change there. And secondly, phenophase changing due to climate variables. Secondly, the potential overlap change. So using the 2 potential overlaps that I explained before, we wanted to see if this potential overlap is changing over time, also considering the species phenology length. And lastly, biodiversity. Our key question, is biodiversity buffering this potential overlap loss? And so to do this, we calculated again potential overlap, but not pooling by site. So we had one matrix per site per year. And what we did was test if potential overlap was changing over time, also depending on the biodiversity of that site. With this, we wanted to consider if sites with higher biodiversity were able to buffer potential overlap loss of species. Just to give you some results, because there's too much to talk about. First one, change of phenophages over time and climate. Indeed, we found that species are changing their phenology over time. They're advancing their phenology, both plants and pollinators. If you see in these graphs, here in the y-axis is the phenophase, and in the x-axis is the year. So we can see that they both have a decreasing trend, so they are advancing their starting of the phenology. However, when we look at the phenology length, we see opposing trends. So basically, plants are behaving as we expected. They're shortening their phenology. However, pollinators are not. For the next analysis, I'm going to focus just on the pollinators, as for plants was not significant. So overall, the overlap, the potential interaction overlap decreased over time. So if we look at pollinators, indeed, we find here the potential overlap and the year, and we see that the potential overlap has decreased over time. However, we see that the baseline of the overlap, it depends on the number of partners that we have. Obviously, if you have more partners, you're going to overlap more days with more species. But what happens if we look at the biodiversity? Doing the same analysis for the biodiversity, what we saw is that biodiversity is not buffering completely the overlap loss. So even though there's sites with more species, these species are not are also suffering from overlap loss. In conclusions, plants and pollinators are equally advancing their phenologies and are mostly driven by the same climate variables. Potential overlap has decreased over time, showing significant trends for pollinators. And biodiversity increases the potential overlap baseline, but it's not completely buffering potential overlap loss. So let's continue with the second chapter. In this one, we want to tackle the process of rewiring in open communities, understanding species turnover depending on the surrounding habitat. In this chapter, the main objective is to focus the species turnover and interaction rewiring, considering the species habitat preferences along a land use gradient. So basically, if we have, again, our network example, if we consider this one as a network that we might find in 100% natural habitat in the surrounding area, what we want to see if there's a change, if we change the percentage of natural habitat, for instance, and also how these species abundances are changing depending on this surrounding habitat. So to do this, what we did was collect data in 2022 of 30 sets and 4 rounds. This was also done under pine forest and shrublands. And we also created these maps of land use in a buffer of 2 kilometers around the site. For the analysis, first we plan to analyze habitat preferences per species with a dataset that is independent from this one, that is for the Iberian Peninsula. Secondly, a landscape composition analysis to introduce not just the habitat percentage, but maybe other variables in terms of landscape, like crop heterogeneity and patch connectivity. And lastly, and the most important question, to forecast the species turnover and interactions rewiring depending on species habitat preferences. So what we expect is to find, if you see in these different plot sites, we have here a very heterogeneous crop, well, a more agricultural land site, and here a more natural land site. So if we take the species abundances, what we expect is that each species with its preferences will have a different abundance depending on the habitat that we see in the buffering area. So our objective will be define these curves and define when can we find each species and how abundant it will be. And secondly, so if we change the species, we might also see new interactions come in. So if this is our interaction network and this is our interaction network for a more agricultural side, what we also want to do is to study these new potential interactions and all the potential interactions that could happen in that habitat gradient and species abundances. Thirdly, the next chapter is focused on the impact of spatial heterogeneity when we collect plant pollinator network data. So in this chapter, the main objective is to assess interaction distribution at a fine spatial scale. So what we want to do, we already know that sadly species do not distribute like this. They do not distribute homogeneously, rather more like this. So we can find one pollinator species and a group of plants here. Maybe here we don't find the same pollinator species, etc. So does it happen the same with interactions? Or can we find all interactions at every site if the species are present? Or on the contrary, we see that some species interact with different plants depending on maybe the resources they have in the surrounding area, the abundances, or other factors. So what we plan to do, well, what we did was we did a sampling in 2024, this spring, of 150 quadrats of 100 meters squared, and in 2 rounds. It was really hard work because also there was the mosquito explosion there, and well, but so in total we sampled around 2 kilometers a squared area. And what we want to do now is to assess the spatial distribution of interactions and how these interactions how is the interaction saturation curve depending on the sampled area. So first, we also did a map of floral resources distribution thanks to the last with drone photographs. Next, we have the interaction data per quadrat that we collected, and we want to finally do is these maps of interactions and assess these different objectives. And lastly, the 4th chapter, a bit more theoretical in a way. So we want to predict plant pollinator interactions at the European scale. In this one, our objective is to obtain a probabilistic matrix of interactions for the European plant pollinator meta-web. So this means that if we have the whole species list that we have in Europe and with their interactions that we find in other studies, what we want to do is to obtain the probability of these 2 species, for instance, interacting at the European scale, and also add functional traits and other characteristics to the meta-web, so we can introduce, for instance, new species that are invaders or invasive species, or other new species that are moving due to, for instance, climate change. So to do this, what we are using is the data set that is from 35 countries, European countries, more than 500 plant pollinator networks, and this has created what we call EBD-net. It's going to be published soon, hopefully. It's still in revision by Jose Naluza. And what we are doing now, so using this EBD meta web that is composed by all the pollinators and plants, we use a combination of algorithms called linear filtering and single value decomposition to obtain these probabilities, and in a later stage incorporate functional traits like size, taxonomy, and other characteristics that could add this biological part to it. So the expected results will be to obtain this probabilistic matrix of interactions with the biological information there to predict potential new interactions in a context of climate change and also invasions. For now, what we have done, it's it's not too advanced, but still we're hopeful that it's going to turn out good because we're getting good accuracy. So too good accuracy will be unbelievable. So basically, it's not possible to have a 100% accuracy. So we're very happy to find the first results that are like positive. So just in a summary, in this thesis, I plan to assess plant pollinator networks requiring from different perspectives, and in 4 different chapters. In the first one, I plan to understand biodiversity as a buffer of phenological asynchrony In the second one, we want to study habitat-dependent rewiring and species turnover. In the third one, we want to study heterogeneity of the spatial distribution of interactions. And lastly, we want to do the predictability of plant pollinator networks. Thanks to my supervisor, Nexi Bartomeus, my PhD tutor, Paco Rodriguez, Curro Molina, for collecting all the data, and also my 4-chapter collaborator, Timothy Poisson, that is the one that created the methods that I mentioned, and also Matthew Blue for offering the climate data. Thanks also to the people that collaborated with the 3rd chapter sampling, because it was really hard and we needed a big team. And lastly, the funding organizations, and just thank you for your attention.
Thank you very much, Nerea.
Thank you.
Time for questions.
That was a very nice talk. Your
Thank
slides
you.
are very beautiful.
Thank you.
I had a question about your first chapter when you were talking about biodiversity. I was wondering how you were measuring that.
It's just number of species in a site.
Okay. Do you think your results would change if you took into account frequency?
Probably. So also biodiversity in this case, it's just the number. So for this 27. For the 37 species that we are using in the study, we're collecting all those 37, and this is the biodiversity we're considering. So probably taking into account frequency might help, but at the same time, we don't have frequency data per se. We have maybe flower abundance, and pollinator by individual, but that's very variable, and some species we're only seeing once. So this was the easiest way, or like the less tricky way to test it. It has a lot of, it will need a more specific fieldwork to test properly how phenology is changing and how biodiversity can change. Because also there's a lot of, there's some papers that suggest that also introducing abundance of the species that you're estimating the phenology will help to better work with this. So it's a bit tricky, but we wanted to do a first approach to it because we had a really good data set, but it's not a specific, because it wasn't planned for this, so we have a bit of we had to work with it. Thank you.
Yeah, over here. Yeah, thank you, Nerea. Excellent presentation, by the way. So I've read this week a very interesting paper. I think the note was on The Guardian, that the phenological adjustment of many plants in different biomes is slowing down over time. And it makes a lot of sense because any organism has physiological limits, things can do and things that can't do. So I've been thinking about this reading during your presentation. Are you considering in a way that plants and insects have this kind of physiological limits that if warming keeps pushing, these actors can simply disappear in suddenly, actually?
Yeah, indeed. So there are some papers that study, for instance, for pollinators, for bees, how bees, so a combination of bees that are already adapted to warm environments and how they could adapt to climate change. So they see that because they're already warm, they reach the thermal limit really quickly, so they die. So basically, they cannot adapt more, what you're mentioning right now. So the thing is, we're not sure. So we haven't tested thermal limits here. We don't have the data for all the species that we're treating here. It would be really nice to do that. Also, we might not have enough enough long time series to test this, because maybe we had. Always, it's better to have more data, you know. Like maybe we had 20 years of data, we will see this slowing down of the adaptation to climate change. I haven't tested how, like, if they're reducing the speed of change, to be honest. But compared to other studies that they have done at European level, they seem to be adapting pretty quickly, because they changed. So these pollinators and plants, they're advancing around 1 day every 2 years, their phenology, and other studies have seen that is 1 day per decade, for instance. So we're still, I think, in a fast moment now. No worries.
Thank you, Nerea, for the great talk. I have a couple of questions. The first one is somehow a follow-up of Jennifer's regarding how to incorporate biodiversity in the model of trends of interaction richness. Are you planning, because I think it would be relatively easy, are you planning to incorporate, rather than just testing the effects of number of species, analyzing those trends for different functional groups of pollinators? Just grouping the species within functional groups and then looking at the trends, not of the specific species, but of the functional groups.
Yep.
Then the second one, very quickly, is regarding to the last chapter. The question is whether the algorithms that you are using to estimate probabilities, interaction probabilities, also yield measure of uncertainty, levels of uncertainty associated to those probabilistic estimates.
Yep. So thank you, Pedro, for the questions. Going for the first one, we have tested that. And so we have tested if yield is, there's a change in yield. Because first, when I started this analysis, I wanted to reduce the complexity, as we don't have a huge data set. We have a really big data set, but not a huge data set. So when we studied at the pollinator, at the species level, we couldn't add as many, for instance, the random slope to see if they're changing differently and stuff like that. So we had to simplify the model. So I tried to do it at the yield level, and we didn't find any signal there. So that's why we went back to the species in the analysis. And in the second part, yeah, it has an uncertainty measure too. So it will be both the probability and the uncertainty of that probability. And usually the model, so the algorithm, for what I understood, because it has a really big complex matrix algebra behind. So for me, I'm still trying to process what it does, but if it reaches a certain, so 0.5, it's certain, so it's going to balance the probability also by the uncertainty of the probability. So it's going to give both results. We're going to know if it's uncertain or not, and what's the probability of that interaction.
Thank you for your presentation. I didn't understand why or the reason you build the probability matrix for the entire meta web. Is it for taking samples for that meta web and predict the network at a local scale? And in that case, have you thought about the potential changes in the probability values once you take a sample for the entire meta web?
So this is like the previous step of that. So in this case, what we're doing is from the lower scale, so from the study scale, we collect the meta web. And our objective with this meta web is to work with a European project that is building climate models and different climate scenarios. So if we introduce, this is why we want to introduce the functional traits, and so with thermal limits, size, movement, and other factors. So we can predict how species will move around Europe and how this will change the interaction networks around Europe. Indeed, the next step will be to size down and being able to get the probability of interaction in a smaller scale. We are still testing that because we're not sure if it's going to be good enough. Of course, as you mentioned, there's so many things going on at the smaller scale, but we expect that it's going to work, because for now we're working with both the meta web and the country level meta webs, and they're giving pretty similar results. So maybe we can indeed downscale what we have in the meta web, the probability, and work it at the study site, for instance, or at ecoregion or even so. It would be great to transfer these for policy, for instance. So that's why we're testing different sizes or different scales of the MetaWeb to then later being able to apply this to also policy.
Okay, is there another question? If not, thank you very much for your attention. Thank you, Nerea, and thank you, Adrián, for your talks.